Good morning to everyone who have connected with us today in the live streaming. Water is life's matter and matrix, mother and medium. Water is the most critical resource issue of our lifetime. Its health is the principal measure of how we live on the land. On this memorable day of imparting and exchanging knowledge, I take the opportunity to extend to you all a warm welcome to this national virtual seminar on waterborne pathogens, the never-ending threat, organized by the Department of Microbiology, Auxilium College. It is indeed a great honor for us to have in our midst Dr. S. Joseph Christian Daniel, Associate Professor and Head, PG and Research Department of Microbiology, St. Joseph's College of Arts and Science, Cuddalore. Sir completed his undergraduation in zoology and post-graduation in microbiology at Sri Paramakalyani College, Alvar Kuruchi. His MPhil biotechnology at Bharatidasan University and PhD in microbiology at University of Madras. He has a teaching experience of around 25 years in the field of microbiology. Counting onto his membership in academic and professional bodies, he is the chairman of Board of Studies in Microbiology, Thiruvalluvar University, Vellore, life member of Indian Science Congress, executive committee member and life member of Indian Association of Applied Microbiologists, life member and Association of Microbiologists of India and Indian Virological Society, editorial board member of St. Joseph's Journals of Humanities and Science, associate editor of Indian Journal of Applied Microbiology, coordinator of Research Development Cell and Internal Quality Assurance Cell of St. Joseph College of Arts and Science, Cuddalore, and a member of Inspection Committee for granting affiliation to colleges set up by Thiruvalluvar University. Sir has received Outstanding Teacher Award for the year 2016 from the Indian Association of Applied Microbiologists. His research area of interest includes tribal health, environmental microbiology, curriculum development, quality assurance and enhancement in higher education. Dr. Daniel is the co-investigator of the ongoing research project, soybean milk fermentation with lactic acid bacteria for nutritional benefits, an intramural research project funded by his mother institution. Principal investigator of completed research project in curriculum development for capacity building in disease modeling, a HADM small development grant project, award provided under the US NIH Fogarty E-Capacity Grant for empowering Indian health researchers with computational modeling tools, a collaborative education project being conducted by the University of Pittsburgh, Indian Institute of Public Health, Hyderabad, and Share India with a grant budget of $5,000, and co-investigator in the ICMR ad hoc projects under the tribal subplan estimation of hepatitis disease burden among Irula tribes of Tamil Nadu and Puducherry. Adding to his credit, he has published research papers in various national and international journals of repute, authored 3 books and 2 book chapters. Sir has attended and organized several symposia, conferences, seminars, workshops, and training programs, and has presented papers. He's an invited resource speaker in conferences and has also chaired different scientific sessions. Sir, we truly value and appreciate your presence in our midst today. I, on behalf of the Department of Microbiology, Auxilium College, and on my personal behalf, extend to you a very warm welcome. I wish to extend my hearty welcome to all the participants from various colleges and universities in and around the nation who have connected with us during this pandemic, and I thank all of you individually for finding time to stay connected with us today. Sir, the scientific forum is yours. Over to Dr. C. Daniel.
Thank you, ma'am. Thank you for the nice words. First of all, I express my thanks to the organizer, Auxilium College, especially the Department of Microbiology, its HOD, Professor B. Gauri, other staff members of this department, and viewers online. A very warm good morning to one and all. And today's topic is waterborne diseases, and the pathogens involved in that will be explained. And I will go for my presentation with your permission. So the topic is waterborne diseases and the pathogens of this particular disease group.
Sir,
The
sir,
never, yeah, please.
your screen is not visible, sir. You have to share your screen.
Yeah, is it available now? I have
No, no, sir.
shared it.
No, no.
Just check it a few minutes. Is it visible now?
No, sir.
Let me check. Okay, sorry for that. Is it visible now?
Yes, sir. Yes, yes. Yes,
I
sir.
am on
Yeah.
full screen mode.
No, it's yes, yeah, of course. You have to hide that screen sharing, sir, actually. Yeah, you hide it.
Okay,
Or you can minimize. Yeah, yes, sir.
fine. So, waterborne pathogens, the never-ending thread, will be the topic. So, everybody knows the importance of water, which has been usually described as the elixir of life, because it is indispensable for not only human being, any life form on earth, and it is a very essential composition of our body, making 70% of the body is water. It is making the composition of our tissues, transports diverse substances throughout body. And like a school child, we will be knowing what are the uses of water, a wide range of uses, household uses, agriculture, energy, medical, industrial, and recreation. It's an endless list. So before actually going into the exact topic, why these waterborne pathogens are considered as a never-ending threat. So you might have read in various reports, newspapers, all social media, water scarcity is a problem throughout the world, and there are a lot of effort taken throughout the world, but still billions of people are lacking safe water, sanitation, and hand washing facilities. So once water is not available, it is not just the water for drinking is not available. It is the water for sanitation and hand washing facility, which is very essential to prevent most of the infectious disease also not available. What's the reason? There is a huge demand for the water everywhere, and the water security is being threatened by so many issues. We will be seeing that. And there is an increasing frequency and severity of droughts. One part we have droughts, lack of water, other parts floods, excess of water, and this is mainly due to the climate change. So these are the reasons why still people are unable to get water for their usage. There are some reports which are available from the UN report. So it says in the year 2015, a report was submitted that says 785 million of people are still lacking basic drinking water facility throughout the world. 785 million people are not able to get good drinking water. That is an United Nations report as on 2015. And another report in 2016 from the United Nations says one third of this primary school throughout the world is not having basic drinking water. water for sanitation and hygiene services. Just imagine, throughout the world, one third of the primary schools is still lacking water. And another report in the year 2017 says 3 billion people are without basic hand washing facilities at home. We are in a pandemic where hand washing is stressed, but that report in 2017 says 3 billion people are without basic hand washing at home. Of course, there may be some improvement in a couple of years, but still it is a problem. So before going into that one, I would like to introduce some terminologies. Of course, we know all these terminologies already, but still, let us be clear with the terminologies. So first one is the waterborne diseases, or sometimes it is referred to as water-related diseases. Almost they are used interchangeably. Any disease which occurs due to the consumption of water or usage of water is called waterborne diseases or water-related diseases. Of course, we being most of us are microbiologists, we concentrate on the waterborne infectious diseases, but there are other diseases which are also due to water that because of the problem of contaminants, chemicals in the water. I'll just tell you two examples. One is a radon, which is a byproduct of decomposing uranium, which produces a radioactive material radon, which is known to be a carcinogen. And another one, this is another serious problem throughout the world, arsenic, which is also found in water as a contaminant. And again, it is causing a collective term of disease called arsenicosis, which is also having some effect on the brain, eyes, and it is also leading to cancer. So these chemicals are also responsible for the waterborne diseases, but being microbiologists, and as of the talk goes today, we concentrate on a sub area of these waterborne diseases or the pathogens involved in that is called water-related infectious diseases. So we'll focus on the infectious agents alone. So these waterborne pathogens are the microorganisms which are known to cause infection in human beings. And this term, water-related infectious diseases, can be further grouped into two. This is based on a reference. You can see it on the screen. According to Batterman, it is two types of diseases when you study the water-related infectious diseases. One is a waterborne infectious diseases, where the microbial pathogen or microbial agent is directly responsible for the disease, waterborne infectious disease. To take the example of diarrhea, due to the poor sanitation, inadequate hygiene, ingestion of contaminated water or water with microorganism, you get diarrhea. That is coming under the group waterborne infectious diseases. The another group is the water associated vector-borne diseases. Mostly we won't concentrate on this, but still water associated vector-borne diseases are very important because certain pathogens of microbial origin, they are transmitted through vectors, vector-borne diseases, and these vectors actually require water for their multiplication. So once this water is available, these vectors multiply there and transmit the disease. You have two groups of examples here. One is a malaria and dengue fever, which is transmitted through mosquitoes, which usually breed in lakes, lagoons, ditches, containers like pots and tires. This is one group, and another group is the schistosomiasis, which is caused by a worm known as blood fluke. The blood fluke needs snails for its life cycle to be completed. So this also is mainly because of the contaminated water, because snails need water, and from that water and from the snail, this blood fluke is transmitted to human being, causing disease. So we have the two terminologies, waterborne infectious diseases and water associated vector-borne diseases. Both are very important. So let us quickly look at what are the pathogens of microbial origin which are important in public health concern. I'm not going into the detail of all these microorganisms because most of us will be familiar with all these pathogens, but just I will list it out and then move to the next part. So you look at this one, we have the causative agents, the pathogens, bacterial pathogens, which are transmitted through water and which will be causing diseases like gastroenteritis. You have a long list of bacterial pathogens which are causing gastroenteritis. It includes the Escherichia coli, Salmonella, Vibrio, and so on. And Vibrio is also important because it is causing a typical disease called cholera. And again, the Salmonella serotype typhi is also causing Hendrick fever or typhoid fever. And when you continue the list of bacterial pathogens transmitted through water, we have Shigella, bacillary dysentery is caused by this organism. Mycobacterium marinum, a species of Mycobacterium group. It is also causing an infection, cutaneous infection, and transmitted through water. And we have other pathogens like Vibrio aeromonas, may be responsible for wound infection and septicemia. Then we have Legionella, which is causing respiratory disease, and Leptospira, which is causing systemic disease. All these bacterial pathogens are able to survive, multiply in water bodies, water, and when consumed by the human being or when used for various purposes, they transmit these pathogens and results in waterborne diseases. This bacterial group. If you look at the parasitic group, it is also very important, and most of the waterborne diseases throughout the world and most of the waterborne pathogens which are reported is mainly the parasitic group. When you compare all the three groups like bacteria, parasite, and virus, parasites are the main group of microorganism which is causing problem in healthcare sector. So you have a very famous example of Entamoeba histolytica, known for the amoebiasis. You have the Cryptosporidium. There are two species, Cryptosporidium hominis and Cryptosporidium parvum. Actually, Cryptosporidium hominis is the one which is causing cryptosporidias in human being, and Cryptosporidium parvum is usually found in animals, especially cattle, but that can also cause infection in human being. And another famous example of parasite which is transmitted through water is Giardia. And we have less known organism but very serious organism like Cyclospora, Dragonculus, which are actually equally important in causing diseases. And another waterborne parasite, Acanthamoeba, which is mainly causing keratitis, that is a serious issue. Toxoplasma gondii and Microsporidia. These are some of the parasites which are responsible for causing diseases. And if you look at the virus group, again it is a large group. Mainly the group of viruses which cause gastroenteritis is a major problem in developing world. Most of the poor countries, they suffer due to gastroenteritis, especially affecting the children. So the first known or best known example is a rotavirus, group A rotavirus. Then you have other viruses like norovirus, sapovirus, Hendrick adenovirus, astrovirus, and so on. And apart from this group, the enterovirus is also causing diarrhea, and hepatitis A virus and E virus, which causes hepatitis, is also transmitted through water. And another virus which is being eliminated is the polio virus causing polio. So this is just a list of waterborne pathogens, which is very serious. And now we look at what are the sources of waterborne pathogens, from where we occur these waterborne pathogenic infection. So let us look into the details. Of course, it is everybody knows, but we won't give importance for these sources. First is the water which is used for the drinking purpose and cooking purpose, household use. And other water which are used for the hygiene purposes, these are the main source of waterborne pathogens. Of course, the source of water may be surface water or groundwater, as we know, and surface water include lakes, rivers, ponds, streams. And when you compare the surface water and groundwater, it is easily understandable that this surface water can be contaminated easily because they are open and having larger surface area. But when you look at the groundwater, it is sometimes classified into shallow groundwater, deep groundwater. And in both the cases, compared to the surface water, they are less polluted or having less number of microorganisms or waterborne pathogens. And sandy soil, when you consider the groundwater, this will allow rapid migration of water. So once the water is flowing into the soil, automatically the pathogens can also enter because sandy soil is having some spores which can easily allow the water as well as the pathogen when you compare with clay soil which is densely packed. And shallow wells, natural springs, they are also contaminated by the surface runoff. When there is a flood, when there is some water from the drainage pipe is flowing on the ground, and if it reaches the shallow wells, there is a possibility of this groundwater being contaminated. And same way, rainwater which is washing out all the waste, that also contaminate these shallow wells. They may be contaminated by dead animals, soil, and other. And another group of sources is the animals and people who used to take bath in open water bodies. That they contaminate. Sometimes defecation by the human being as well as animal is a major source of contaminating water, and they may cause diseases also. And another source of waterborne pathogen is rainwater. Of course, when there is a scarcity of water, people used to collect rainwater when it rains. Of course, rain is not so frequent now, but still, this rainwater is often collected from the roofs and stored for use in a later stage during dry season. And when it is collected, if it is not properly collected, this rainwater, we are not talking about the rainwater harvesting where the rainwater is allowed to go into the earth and stored there for groundwater. We are talking about the rainwater which is being collected for various purposes. So if it is not properly collected, it may be contaminated by the birds or animals which defecate on the roofs. It may be plant leaves and other soil particles which are dirt particle, any waste which is found on the roof or in the open terrace, that may be accumulated and it will be collected along with the rainwater. That may be a source of waterborne pathogens. Especially when the water is not pure, there are a lot of outbreak due to rainwater, especially in case of E. coli, Salmonella, Cryptosporidium, and other pathogens. Then open defecation is contributing a lot because that is a major source of waterborne pathogens, especially gastroenteritis group. Feces as well as urine from the human waste as well as from the animal waste that may contribute for this waterborne pathogens in the water bodies. And rarely hospital waste, when it is not properly disposed of, when it is not properly treated, that also contribute for the waterborne pathogens. Look at the other group of sources. This is when compared to the consumed water for consumption. When you compare that one with this one, water for sports activities. So this is less dangerous, but still it is a problem, mostly in Western countries. So this waterborne transmission of this pathogen is associated with swimming pools, and we have a term called hydrotherapy, where water is used for the treatment. So that hydrotherapy pools, of course, that is not very popular in our country, but in some Western countries, hydrotherapy pools are very popular, where people get treated for some diseases. So that is also a source of waterborne pathogens. And again, this is also in some countries used for birthing pools. So delivery of the baby in water containment, that is also a reason for waterborne pathogens. Air conditioner, and humidifying devices. These can also contribute for the waterborne pathogens. But when compared with consumed water, household water, it is less. And endoscopy water is also is treated as a major contaminant when that washer is not properly disinfected. Water used for medical purposes, when it is not properly disinfected or properly maintained, the containers, if it is not cleaned frequently, that may also allow the growth of waterborne pathogens and may transmit this to human beings. And organisms which are normally coming under this category include gram-negative bacilli especially, and mycobacteria of environmental group, and Legionella species. And the next category of source of water is ingestion of water, when it is contaminated with the female feces of animals and human. That is also your source. And usually discharge from the municipal sewers. When there is a drainage system, if it is not properly disposed of, or when there is a damage in the pipes, municipal sewers, that may contaminate the water sources. That is how it is contributing for the waterborne pathogens. This is another serious issue in countries where the sanitation facilities are not proper. And municipal sewage contains human feces because it collects all the fecal matter from the houses and collect a common pit and dispose it up properly. But there is a possibility of leakage and there is a possibility of disposal of this sewage without proper treatment. That is also contributing for the waterborne pathogens. And mostly this will lead to the waterborne diseases. So next we will look at an important concept called waterborne pathogens and indicator organisms. So what is this indicator organism? So let me explain this concept of indicator organism. So when you feel your water is contaminated or containing pathogens, you want to find out whether the water is containing any pathogens, whether it is good for human consumption. In that case, you want to look at whether it is containing any pathogens, but routine isolation of pathogens from any water source is not feasible. There are several reasons for identifying the pathogens or isolating the waterborne pathogens in the water bodies or any water source. One important reason is these pathogens are usually present in small numbers when you compare with other types of microorganisms. Because not all microorganisms in the water is pathogenic, only a few are pathogenic. So when you compare the number of pathogens in the water and non-pathogens in the water, the total microorganism, the total microorganism will be large in number, but the pathogen will be small. So isolating this pathogen specifically is very difficult, one reason. The second reason is there are many types of pathogen, of course not too many, but you have some pathogens which are frequently found in water bodies or water, and you have to use different methods to identify that particular pathogen. So you need different techniques, multiple techniques to identify different kinds of pathogens present in the water. So this is also not possible. For these, because of these two reasons, routine isolation of pathogens from the water is a little bit difficult. So what is the alternative? How will you check the quality of water? So we come to the concept of indicator organisms. So you just look for some microorganisms which are there in the large number in the human or animal gut and often released into their feces. So if you are able to track these organisms which are not causing disease but still present in the gut and released in the feces, if you are able to identify these organisms, these can be used as an indicator for the presence of pathogen, a possible tentative indication of presence of pathogens in the water. This is very important. We are not looking at the pathogens directly. Instead, we look at the source of contamination, whether there is a possibility of feces of either from the animal origin or human origin get mixed up with the water bodies. So that is why these organisms are called indicator organisms. So once you look at the indicator organism, you can suggest the water is polluted or contaminated and not for usage. So we look at this long list of indicator organisms. There are several indicator organisms proposed by various bodies, including WHO. So some of them are listed here: coliforms, thermotolerant coliforms, Escherichia coli, fecal streptococci, and so on. Even the Clostridium, sulfate-reducing bacteria, they are also listed as indicator organisms. We will quickly look at the three important groups of indicator organism. The first one is a coliform organism, or sometimes it is called as the total coliforms. So what is this group of organism? This is the most widely used indicator organism for detecting the pathogens or fecal contamination of water bodies, because it is easy to detect them and enumerate them in any water sample. So the coliform is a group of gram-negative organisms which are having the ability to grow in the presence of bile salt. This is under laboratory condition, because once you are saying or designating an organism as an indicator organism, coming from the source of human intestine, definitely it should be able to tolerate bile salt. So that is another one of the character which defines this indicator organism total coliforms, and it may be inhibiting other surface active agents also, and in their presence, the presence of bile salt and other surface active agents, it should be able to show growth in the laboratory condition. And the main point is it should be able to ferment lactose, because any organism which is residing in the gut of either human being or animals have the ability to use or ferment lactose. So that is a key characteristic. That is why it is called coliforms. At a temperature of 35 to 37 degrees centigrade within 24 to 48 hours, either with the production of acid or acid and gas. So this is a definition for coliform organism, which are used as indicator organism. Other characteristics are oxidase negative and non-spore forming organism, and it will display the beta galactosidase activity, which is connected with lactose fermentation. So the organisms which are coming under this group include Escherichia, Citrobacter, Enterobacter, and Klebsiella. So this is a major group. Usually we look for these organisms in the samples, water samples, when you want to say whether your water is pure or can be used for food or food industry or human consumption as drinking water. So the second group is the thermotolerant coliform bacteria, which is slightly different from the previous one, but exactly some organism from the previous group is subdivided into this group, thermotolerant coliform bacteria. Only major difference is the temperature for the growth. There it is 35 to 37 degrees centigrade, but here it is 44 to 45 degrees centigrade, slightly higher temperature than the previous group. That is why they are called thermotolerant coliform. Of course, not all coliforms will be able to survive this temperature, but a few can survive. They are listed as thermotolerant coliforms. Why this particular group is included is mainly because these organisms can be found in the industrial waste, that is decaying plant materials or soils which is contaminating the water, especially from the industrial effluent, may have this organism. Because of the industrial process, it will be able to survive at a little bit of higher temperature. That is why this thermotolerant group is listed as an indicator organism. Then the third group which I am quoting here is the fecal streptococci, like the coliform. Some streptococci under the genus Enterococcus and another genus Streptococcus are listed as indicator organism. They are collectively called as fecal streptococci, as it can be found in the feces of both humans and animals. And they are also having the ability to grow in the presence of bile salt. So that is why they are listed here. So we have seen three important groups of indicator organism. Now, how to detect this indicator organism? We are not going to directly detect the pathogens, waterborne pathogens in the water. Instead, you are going for detecting the indicator organism, which indicates there is a possibility of fecal contamination and thereby a possibility of disease-causing waterborne pathogens presence in the water. So how will you detect this indicator organism? So fecal contamination can be easily monitored routinely when you are looking for the indicator organism. So this can be carried out by three important techniques. Of course, these are the conventional techniques or the traditional techniques have been developed for so many years. I mean, have developed earlier and still in the use. The three important techniques I will just quickly explain. The first one is the famous MPN method. Most of the undergraduate students or microbiology students will be familiar with this experiment. One important thing about this experiment is this will be done in the practical class, but it will not come for the exam. That is how the students used to look at this experiment, because it needs a lengthy period of time for getting the final results. So multiple fermentation tube or most probable number technique is age-old technique, widely used. Still it is used. So you take a small quantity of water, then distribute in three different concentration of medium at three different concentration of the or volume of the sample, then look for some reaction of the coliforms, especially the color change due to pH change or gas production. Acid production and gas production can be tested in a series of tubes, and you have a mathematical statistical calculation which will give you based on the present gas production or color change and absence of color change and gas production. It will give you a statistical value that can be quoted. This is the famous MBN method. Of course, it is a lengthy procedure but interesting procedure. The second method widely used for the detection of indicator organism is a membrane filtration technique. This is a little bit better than the previous MPN method because there you need a number of tubes to carry out the experiments, but this is comparatively simple. So a measured volume of sample is always taken and you use a filter, bacteria retaining filter of 0.22 micron, then filter the water content and you will be able to filter it out and allow this organism to grow on a medium and count the number of colonies. It is viable count. In this experiment and in the previous experiment, both are viable count. So it is comparatively easy to directly count the number of viable colonies on the medium. And depending upon the type of indicator organism you are looking for, you can select the medium and do it. Then the third method is a very simple method, which is called presence absence test. It is slightly similar to the coliform MPN technique, but we won't be using multiple tubes. We are not going for a quantitative results here. That MPN technique is a semi-quantitative technique. Membrane filtration technique is a quantitative technique, and this presence absence test is a qualitative technique. So this will be mainly suitable for testing the water quality, drinking water quality, when you are not expecting the positive results. So if you are not very particular about the exact number of organisms, you can go for this method, positive absence test. And this is having another drawback. It is not recommended for the countries where contamination is frequent. If the contamination is frequent, if there is a possibility of waterborne pathogens present in the sample, you cannot go for this. But slightly, you can just have a qualitative test whether the sample is having some pathogens present or not. So that is one thing, and it cannot be used for large water supply because if you use a large water supply and take a small quantity of water, you may miss the contaminants or you may miss the pathogens. So you will not be able to get the result. So these are the three widely used methods. Now we will go into the actual topic, waterborne pathogens detection. So the previous one says instead of detecting directly the pathogens, you use a indicator organism and check it. But here you are not going for the indicator organism, is straight away going to the detection of pathogens. Of course, a lot of methods are available. Why this detecting the pathogen is important? Because just now we have seen indicator organism is enough to indicate there is a possibility of presence of waterborne pathogens. But why you have to exactly look into the waterborne pathogens in the water sample? So when you want to ensure the safety of water used for human consumption or any purpose, the presence of pathogens has to be monitored. You have to exactly find out whether the pathogen is present or not. And if they are present, then what you have to do is you have to look for the source of contamination. So indicator will have an idea whether the water is polluted or contaminated, and pathogens once found in the water sample will say that water is not useful for consumption or any other purpose. And the second part is once you detect it, you have to find out whether there is a possible source of contamination. Because unless and until you find out the possible source of contamination, you will not be able to rectify it. So that will be an added advantage when you are going for exact detection of waterborne pathogens. And there are different methods are available, specific detection methods, and you can test the live pathogens from the water samples. And there are some reports available that indicator organism may miss the pathogens if it is large water bodies are used, or if you take the sample from large water bodies, because the possibility of getting the organism will be less. That is one reason. And another reason is there are some papers There are some reports, research reports available that says there is not much correlation between the presence of pathogens in the water sample and the number of indicators pathogens as an indicator of bacteria in the sample. There is no proper correlation. So you can get a false negative result. When you take a sample and look for indicator organism, you may get a negative result and you will consider the water is safe for consumption or use, but it may still contain the pathogens because the pathogens will be always less in number compared to the indicator organism. So when you have an idea of pathogens being expected, first test for the indicator organism, then look for the actual pathogens. That is why pathogen detection is more important than the indicator organisms. Then now looking into that one, what are the challenges? Okay, still you can go for waterborne pathogen detection in any water sample, but why it is not that much easy? What are the challenges when you are going for this type of detection? First one is the screening of water sample for the presence of pathogen is difficult because collecting large amount of water is not possible. Because if it is, say you are checking a water body, larger water body like lake, which supply water for huge population, you cannot just like that take 100 ml of water from that one and say whether there is any possibility of fecal contamination or presence of pathogen. So compared to the huge volume of the water reservoir or water body, the sample which you are going to collect is very less. Of course, you'll be collecting liters of water and from that you will take 100 ml of water and test it. But still, the collecting large quantity of water is not possible. So that is one challenge, volume which you are going to test. Second, how to collect this sample. This is another problem because if you want to detect a pathogen, you have to collect the water properly so that you will not be missing out the pathogen. If you miss out the pathogen in a large water body, your results will be negative. So how to collect the sample is one challenge. Then another challenge is where to collect. Just imagine it is a very huge water body. You cannot collect the water near the edges of the water body because water in the edges of water bodies will be having more number of microorganisms than the deeper regions or middle region. So where to collect and when to collect. This is also very important. The time record for the culture-based study is another problem. How to collect, where to collect, when to collect is one problem. Second is, when you are just collecting the sample and processing in the lab, you will not get the result immediately. See, for example, when you have a culture-based method for a pathogen or indicator organism, it will require minimum of 2 days to maximum of 7 days. And especially in the case of Salmonella, as per US FDA document, it says 7 days you have to wait to get the final result. So this is not always practically possible. This is another important challenge in the detection of pathogen. When you have an outbreak of salmonellosis or typhoid, you cannot wait 7 days to confirm the results and find out the source of pathogen. So that is a problem with this detection of pathogens. So this may have, making the test is financially not possible and logistically also it is not possible. So suppose you are doing the test on a large scale. Then the last point in the challenges in detecting the waterborne pathogen is the occurrence of pathogens will be very difficult or very small in quantity when you have a sample like a small sample or which is less contaminated. So suppose if the sample is having collected from a sporadic case area where 1 or 2 cases have been reported, from that you are collecting a sample, sporadic incidents. So there is not much possibility of getting a positive result because a sample will be having a diluted number of microorganisms or pathogens, but still there will be a threat. So that is one reason. Then heterogeneously distributed. Suppose the fecal matter which is contaminating the water body will be having more number of pathogens, but once this get mixed up with the water body, there is less possibility of isolating the organism because it is getting diluted out. So these are some of the challenges when you are going for detecting the pathogens. So next, I will just give you one example of detecting a pathogen. So it is an old document, but still a valid protocol for the isolation of Salmonella. Of course, the name is given as Salmonella typhi in the old nomenclature, but now it is called Salmonella enterica and the serotype typhi. So this document from the US EPA in the year 2010 gives a detailed procedure. I will give this as an example because most of your students who are interested in experiments. Of course, this can also be used for research, but this is a basic procedure how to isolate a pathogen. So I am taking this organism as an example. So very simple is first one is analysis for a qualitative analysis. You're not going to exactly find out how many organisms are present. You want to detect or you want to find out whether the sample contains a Salmonella species. So what you can do is you can go for a qualitative analysis, take sample, and in most of the cases, the water sample to be collected will be large. Say, for example, depending upon the water source or depending upon the size of the water body, you can collect 5 liters of water or 10 liters of water. From that, you will be taking 100 ml for the analysis. So take 100 ml. Usually, this 100 ml is convenient for calculation and reporting. And this 100 ml of water sample will be mixed with equal volume of medium, double strength medium. Of course, that sample size and the quantity of organism expected to be in the sample, we don't know. So we will be tentatively using a double strength medium. You will know how to prepare a double strength medium. If a particular quantity of nutrients has to be added to 100 ml, if you double the number of or quantity of this medium, that is double strength. So you prepare a double strength medium. Any medium, of course, here it is called universal pre-enrichment medium or universal pre-enrichment blood. But according to the regulations available in different countries and different agencies, you can select the enrichment medium. So this is actually an enrichment technique. Double strength is used because the double strength medium will compensate the nutrients when you mix 100 ml of sample with 100 ml of broth. So it will become 200 ml total quantity, and the double strength medium will now come in single strength, that is 1x. And incubate this at 35 degrees centigrade for 24 hours. Then after that, you can look at the results. Depending upon the medium you have used, either you will be looking for the turbidity in the medium, or if it is having some dye and some sugars, you will be looking for a color change. So after 24 hours, you just look at the culture where you inoculated the sample, and depending upon the color, you can find out whether there is a possibility of the organism being present or not. This the first step. So then you have to go for a selective enrichment of the salmonella, because this protocol is mainly for the salmonella. So we use medium and procedures only for salmonella. Suppose you are looking for some other pathogen, for example, Vibrio or Shigella or any other pathogen, you have to select the enrichment medium and selective medium accordingly. So in this case, we are looking for salmonella. So we will be going for the next step, enrichment, actual enrichment using selenite cysteine broth, or simply called as selenite broth, which is a selective broth for the salmonella. Mostly, this broth will inhibit E. coli and Shigella to a larger extent, but there are some cases where still you will be able to get the colonies of E. coli in the medium after taking from the selenite F broth. So you just take the culture which is giving positive results in the previous step, mix it well, and transfer 1 ml from the culture of qualitative sample to the tube with 10 ml of selenite broth. So now you are actually going for the isolation of the organism by enriching it, specifically enriching it. Because the water may contain any organism, heterotrophic bacteria will be there, indicator bacteria will be there, but we want to find out, or we want to detect only a particular specific pathogen which we suspect. So incubate it at 35 degrees centigrade, and after incubation, you will be able to see the color change or turbidity according to the medium you have used. Now you are going to isolate the organism on a solid medium. You want to isolate the colonies. So in this case, since it is salmonella, we can use any medium which is specific selective medium for the salmonella. In this protocol, they have given two medium, bismuth sulfate agar and Miller-Mallinson medium. These are the two medium for the selective isolation of salmonella. So from the positive tubes, you can take a low fill of culture and stick it on these two plates using a sterile, of course, you know the procedure. Sterile inoculation is done and keep the plates at 35 degrees centigrade for 24 to 48 hours. So depending upon the medium, you will be able to see different colonies on the medium having the similar expected colony morphology of salmonella. So if it is going to be salmonella species which is causing typhoid, that serotype typhi, you can get green-black colonies with metallic sheen on bismuth sulfate agar. And if it is Miller-Mallinson medium, you will be getting black colonies. So now, after pre-enrichment, enrichment, then after enrichment, you have isolated the colony. And sometimes, if you have a doubt, if you are not very sure salmonella is going to be the pathogen in that particular outbreak, you can also use some other media side by side. Parallelly, you can use other media because you should not miss out any organism, possible organism. So normally recommended medium in this protocol is tryptic soy agar, or the very common medium which you can use is Macangi agar, because Macangi agar can allow the growth of any of the coliform group. So that will be an ideal medium, or you can go for tryptic soy agar if you are not sure of the expected pathogen. So the same way from the positive tubes of selenite F breath, you streak it on tryptic soy agar and incubate it. So that will be one. Then you can further purify the organism using any of the medium, and the colonies can be stored. If you want to do further examination, or if you want to study that organism further, you can store it in any slant, tryptic soy agar slant, or the widely used nutrient agar slant. Then you can go for the other tests. So this is just how the, of course, everybody knows it is not a novel result. So you have Macangi agar with salmonella and other organisms lactose fermenting, and salmonella is non-lactose fermenting. So the colorless colony will be the what do you call salmonella, and XLD agar, another medium widely used for the isolation of salmonella, and BSC. So once that organism has been identified, isolated, you will be able to biochemically confirm it, and serological methods are also available. Of course, we will be using salmonella anti-serum, VA, that can be used. But now we have so many sophisticated tests available, you can simply go for the PCR. 16S rRNA sequencing can easily identify and confirm whether it is salmonella, and even up to the level of species. So serological methods can also be used, where you have to select a colony and you have to mix it with a saline, make a suspension, and use the anti-serum to confirm it. So this is a simple protocol, age-old protocol. Of course, there are so many novel techniques available, but still, this is what you can easily do in the lab, and research purpose, you can start with this experiment. Okay, now let me go into some of the advanced techniques which are available, which are employed for the detection of waterborne pathogens. So we cannot rely on the older techniques, traditional techniques always. When there is an advancement in technology, though there are some difficulties, still we have to depend on we have to move for the advanced techniques. So this is what you can do is find out what are the novel advanced techniques available for the detection of waterborne pathogens, and standardize it and use it. So it depends upon your convenience, your aim, your the facilities available with your lab, and affordability and the need. So you can select any one of the advanced techniques and use it. So I'm going to just brief a few modern techniques which may be useful for the detection of pathogens, waterborne pathogens, and of course, it is applicable for any organism. These techniques are common techniques can be used for any type of environment. So first method is calorimetric and colorimetric method widely available. So this is product by companies in commercial scale. I'm not recommending any, but I have taken one example of a trademark one from the laboratories, Qualitative Analysis. So they provide kits, ready-made kits available, which can be used to detect these pathogens. So use the technology in conjunction with high sensitivity fluorescence detection. So they designed a kit which will be detecting the fluorescence using an instrument, and this will be combined with a substrate which is specific for an organism. So you have an enzyme from the organism, you add a substrate, and this substrate is actually combined with a fluorophore. So this enzymatic reaction, excuse me, I think I'm hearing some noise.
So here it is clear, actually.
Okay,
There is
fine.
a break in your, yes, sir. Yes, continue.
Okay, so this technique, the colilet kit or enterolet kit, is specific for coliforms. So what this technology does is simply combining an bacterial enzyme which can be used to convert a substrate. Any bacterial enzyme will be acting on some substrate, and if you attach a fluorophore with a substrate, then you will be able to produce a fluorescence, and you can measure the fluorescence using a device available with the instrument that can tell you whether the pathogen or the indicator organism is present or not. So the colorimetric method or fluorimetric method is used. What is the advantage? This can be used to detect the number of organisms in a few hours, 18 hours, say for example. Maximum of 18 hours, you will get a result with the help of the automated instruments. And another advantage with this one is the sensitivity. Since it is looking for a enzyme and substrate like your ELISA, this is very specific. So the sensitivity is high. That is one. But longer the analysis, you will be having better results. So this is one method. Now advanced technique, colorimetric and fluorimetric method. Lot of kits are available from different companies for different organisms. So you can select your organism and look for a kit or look for an, what do you call reagent, get it and do the test. What you need is an reader or instrument to measure the fluorescence. This is one technique. Most of the high-end labs are using this technique from different companies. I have just quoted IDEX, but any company can be used. And second one is a very familiar technique, the modern technique which revolutionized the identification of microorganisms, the PCR technique. Of course, in the COVID period, we are very much familiar with this PCR, RT-PCR. So polymerase chain reaction can be used to detect the pathogen or the indicator organism or any other organism in the sample. You can just isolate this organism from the sample or directly use the sample, separate the DNA and do a PCR with a known specific primer. So you can choose a particular primer, either looking for a particular organism or particular group of organism. You can select the primer accordingly and do the PCR. Okay, so that will give you a better result comparatively. And what are the advantages of this PCR detection of waterborne pathogens? It is rapid, highly sensitive. Of course, we are going for the gene, so highly sensitive and accurate. And another advantage is mainly we are talking about waterborne pathogens in the category bacteria, but this technique is also suitable for viruses and protozoa, because whatever we have discussed earlier is mainly suitable for bacterial pathogens or bacterial indicators, but this PCR can be used for viruses as well as protozoa also. That is the advantage of PCR. Then any technique is with some limitation. No technique is without limitation. So There are some limitations with PCR. So false positive results will be a major problem in this one. When some naked nucleic acids, DNA or RNA is present in the water sample, that may interfere with the result. Suppose if you're not going for a culture, pure culture, if you take a sample and isolate the DNA and look for a particular organism, there is a possibility of DNA present in the water sample may interfere with the results, may give false positive also. Then sometimes, since it is a DNA based technique, you cannot expect always a viable organism contributing for the DNA. You can take the sample and even the non-viable microorganism once have been eliminated or killed, maybe having its own DNA left out in the sample, that can give a false positive result, or there is a major problem, there is a contamination because the water or reagents and the lab setup, everything should be perfect for conducting the PCR. Otherwise, you may get a false positive result or even some contaminants. And another drawback is the environmental contaminants, not just microorganisms. Any particle, any dust, any waste material in the water sample may interfere when you are taking the water sample and do the analysis. So this is the second advanced technique. We'll go for the third one, biosensors. Of course, biosensors are a rapidly growing field. It has found use in so many fields of biological arena. You have use in the medical field, in industries, food industry, everywhere you have now biosensors, and the same biosensors can be used for the detection of waterborne pathogens also. Of course, it is not very new, but biosensors have been developed for a number of molecules. Here, we are not exactly looking at the organism. We are looking at a molecule, either part of the organism or produced by the organism, and you are detecting the molecule. So the specific, something like an antigen or any enzyme or DNA, that can be identified by this or detected by this biosensor. So that is a method. What are the advantages of using biosensor? Highly sensitive. If you are correctly selecting the analyte, that is very important. The analyte which you are going to select for the detection using a biosensor should be specific for that particular organism. You want to look at salmonella or you want to detect salmonella, you have to select an analyte which is specific for salmonella. So that is one advantage. If it is suitably selected analyte, you will have high sensitivity, short time, and high specificity. And still, the technique is having some drawbacks or limitations, especially when you are detecting waterborne pathogens, because the pathogens will be present in very low concentration in a large water body. So you may miss the analyte, or your technique may miss the analyte. So that is one drawback. Second, sometimes if you are going for an environmental samples directly, that sample may contain inhibitors of enzymatic reaction. Suppose in the biosensor method, if you have selected one enzyme as the analyte, if you are selecting one bacterial enzyme as an analyte and you are using this biosensor technique, if there are enzymes present in the water sample, that may inhibit or interfere with your result, or sometimes other enzymes can also give false positive result. Inhibitors of enzymatic reaction is one drawback or one problem you can face. Then interfering organisms and particulates. So if you are looking for an enzyme, that enzyme may be produced by a non-pathogen also. So that may interfere with your results, or any dust particle or waste material present, turbid material present in the sample may interfere. What is very important is how you are selecting the analyte. Most of the techniques use still DNA as the analyte. It is very easy. You can have DNA like the PCR. It is, of course, similar to PCR. So your biosensor method looks for a DNA molecule from your organism, specific DNA molecule or gene, then it can detect. So biosensor is having lot of application in waterborne pathogen detection also. And the last method which I am going to describe is the flow cytometry, and another technique called fluorescence activated cell sorting. Of course, again, these are novel techniques finding use in a lot of field, not only in microbiology, in human cell culture, tissue cultures of plant or animal origin, you have this technique, flow cytometry, food industry. So there are several industries which started using these techniques, flow cytometry and fluorescence activated cell sorting. So this is also used for the detection of waterborne pathogens as a tool. Of course, it is not very new. You can look at the reference quoted there, Porter et al. In 1993. So that is a paper quoting the use of these techniques for the detection of aquatic bacteria. And what is the advantage? It can be used to isolate a single cell directly from the water sample. Even if it is a single cell, you can look for that cell and detect it. That is one. And advantages is it is reflecting the bacterial population diversity. So what is very important is you are not looking at one particular pathogen or one particular indicator. Using this technique, you can find out any number of organisms, bacterial population diversity you can study. This flow cytometry and fluorescent activated cell sorting method can detect a number of organisms irrespective of their pathogenicity. So that is one advantages with this method, but still it is also having some drawback. Difficulty is detecting and isolating the pathogens that are present in low concentration in the water. So if even it can, the main advantage of this technique is it can pick up even a single cell. But just imagine if you have lakhs and lakhs of liter of water in a water sample or water body, and if you take, or say for example, 1 liter of sample or 100 ml of sample or 1 ml of sample, the pathogen or the organism may be missed out. So that is a drawback. Now, the final part of this talk, the waterborne pathogens, prevention and challenges. Okay, now we have a clear idea of what are the pathogens present and from where they are coming, how to detect them. Now, how to prevent the diseases caused by these waterborne pathogens, or how to restrict these waterborne pathogens. What are the methods and what are the challenges that we will see. So how to prevent? Prevention is very simple, because if you think, you can find a lot of methods based on the sources of infection. You can find out the prevention methods. For example, if it is surface water which are easily contaminated with pathogen, the simple method is treatment. You have to treat the water bodies or water at any point of use. So chlorination can be done, filtration can be done. This is mainly to reduce the waterborne diseases. Once you control the waterborne pathogens with chlorination process or filtration process, you can prevent the disease, you can control the pathogens. There are newer technologies available. The previous filtration refers to the common filtration like sand filtration, which are not using fine membranes. But newer technologies are available in the area of filtration, where you can use membrane filtration technique, and that will give you a water totally free of microorganisms. Ultraviolet radiation can be used because it is able to kill most of the microorganisms in the water. Ozonation, that can also be practiced. So these are some of the methods, microbial treatment methods: chlorination, filtration, ultraviolet radiation, and ozonation. They either remove the pathogen or separate the pathogen physically from the sample, water sample. For example, the filtration techniques. Or it may inactivate or kill them. For example, your ozonation method or ultraviolet radiation, which will simply kill the organism or inactivate them. So this is mainly suitable for surface water. If you come to shallow wells where the waterborne pathogens can gain an entry into these shallow wells, so very simple technique. If it is a shallow well, build a wall, simple wall around the opening of that well to some height, so that the surface runoff, rainwater or other wastewater cannot come into the well. So you can build a small wall surrounding that one, and you can prevent the entry of water, contaminated water, sewage or other runoff that can be used to prevent the entry of waterborne pathogens. So you can direct the water, that is, if you can manage that area so that the rainwater or other runoff cannot come into the well, that is one way. And if it is wells which are used for drinking purpose, drinking water purpose, simply put a cover on the top of the well that can prevent fecal matters from the animals or birds from falling into the well. That can be avoided. And one important point is well water should be pumped by electrical devices. Of course, this may not be possible. In the olden days, people used to draw water from the wells or deeper bodies using buckets tied to a rope. But there is a very quite common practice. Still, it is used in most of the part of the country or even in a part of the world. But these buckets have been linked to the source of waterborne pathogens. If the bucket is contaminated, if the bucket is not cleaned properly, that can act as a source of water. Remember, the well water is pure, but once you use a bucket which is already contaminated, having microorganism, that bucket may contribute to the contamination. So that can be prevented easily. Then suppose if it is spring, put a house and that build some small box-like structure covering the spring so that the water can be uh, maintained pure without getting contaminated from other surface runoff. Then second thing, if it is a spring, this is a common practice. If it is a spring, don't collect water from the starting point. From the source of spring source, you should not collect water. You should collect water some 1 or 2 meters away from the source of spring so that you will not be contaminating the main source. This is a simple prevention method. Then animals, human beings should not be allowed to bath in water bodies. They should not be allowed to defecate in water bodies. That is a major risk. So if you are able to prevent it, then it is possible. Of course, our country is taking a lot of measures in this area where you will be just looking for constructing toilets for even people in the villages. So that is one thing. Then the surface water bodies is less polluted more, but you can get water always from the deep well. So how you can do this? If there is a possibility of collecting water from deep well, prefer deep well. Of course, there will be difficulty in collecting water from the deep well because you need an electrical pump to pump out the water. But boreholes, bore wells, and deep wells are preferable when you think of contamination because they are less polluted as we have seen earlier. Then these boreholes can be covered and properly prevented from contaminating water. That is one thing. Then if you are going for rainwater source, so the rainwater source is an alternative for drinking water in many areas when it rains, and rainwater harvesting has to be done properly. You have to use sand filtration. Don't collect the water directly and use it. Collect the water, allow it to pass through a sand filters and collect the water. Then flushing tanks. This is another concept because when it rains, immediately you should not collect. So the water will be first, when there is a first rain, water will be collected in a separate tank called a flushing tank. So after that, after 1 or 2 days or after some time, the rainwater will be collected in a separate tank for storage. So not directly the first water which is collected will be sent to the flushing tank, and afterwards the water will be collected in the storage tank. So this is how you can prevent the entry of waterborne pathogens through rainwater. And another point of used water is the water quality device can prevent the waterborne diseases if you are using point of use water quality devices. So what are these point of use water quality devices? What do you mean by point of use? Where you are collecting the water, taking it for human consumption, there you do some precautionary measures, preventive measures. For example, you can use ceramic filters. Of course, we have most of the filters fitted at home for collecting drinking water. The famous AquaGuard is an example. Ceramic filters can be used. Biosand filters can be used if you are collecting water on a large scale. Then solar disinfection, that is also quite good flocculation method using chemical disinfection. This flocculation combined with chemical disinfection is we're using some chemicals both for flocculation and chemical disinfection. So what is the advantage of these methods? Ceramic filters will allow water to percolate through the filter and physically remove all the dust as well as pathogen. You will get a pure water free from contaminants. Solar disinfection is also good. To some extent, it can kill the pathogens or kill the microorganism by ultraviolet rays present in the sunlight. But the problem is you won't get sunlight always. Solar disinfection is not that much convenient. Then flocculation can be done because flocculating agents, a chemical which is added to water, will allow the dust particles along with the microorganism to form floccules. Then that will settle at a later stage and you can remove it easily. Then sanitation methods are very important. Always we combine drinking water usage and sanitation together because water is not simply used for drinking purpose or cooking purpose alone. Sanitation, bathing, and the toilet use, we are using water. That also contribute to the waterborne diseases because it contains waterborne pathogens. So the best method is sanitation method is very simple, straight away method. Avoid or prevent the human waste or animal waste from entering into the water sources. Feces, urine from animals and human being can be prevented. This sanitation will help you a lot. The simple principle is septic tank. Of course, there are places where you have not just septic tank, common collecting point is there, but in any case, this should be properly constructed, either household septic tanks or a common pit or common collecting points. Drainage system should be properly constructed so that these contaminant waste material should not come in contact with the drinking water source at any point of time at anywhere. So this is one important area where you can prevent. So we have listed what are the methods by which you can prevent the entry of waterborne pathogens. Now, it is not that much easy. There are some challenges you can face in providing a safe drinking water or safe water for human consumption. So what are the challenges we are facing? One is the population growth. The population is increasing. Any government is finding it difficult to provide safe water because of increasing population. That's one. Then urbanization. People concentrate in cities. So the cities are not able to provide water. You might have read in newspaper last year there is huge water scarcity in the cities like Chennai. So urbanization is a problem. Preventing agencies, government agencies to provide good, safe drinking water. Population density, this is another problem. This is different from the population growth. Increasing number of population throughout the country or throughout the world, increasing population density is in a particular area, more people resides and providing a water facility will be difficult. So this actually contribute. When you are not able to provide safe drinking water to people, automatically there is a possibility of people looking for alternative water sources. This is a concept. When the government agency is not able to provide safe drinking water, people have their own way of collecting waters from different sources. So unauthorized or not perfect methods. So that will lead to waterborne diseases by spreading waterborne pathogens. So there is one. Even industries are looking for water, that increases the demand for water. Then climate change, climate variability and climate change. Climate variability is that you cannot have a fixed climate. You cannot say it will rain this month and it will not rain this month, but it will be raining in some other month. Variability, change. This will also diminish the water availability. So you are not getting enough water even in the deep well or bore well. It get dried up during summer. Then quality of the water is also poor. And flooding. Of course, we think flooding is also dangerous. When there is flooding, it actually covers all the water bodies and the drainage, everything get mixed up and it makes the water deep, even the deep wells becomes useless once there is a flooding. So flooding is also having a negative impact on water quality and causes the spread of waterborne pathogens. Drought and drainage, poor drainage and storage system is the main problem. Then advancement, sustainable development is a key concept because there is a lot of development going on throughout the world in industrialization, everything. You have built a lot of dams, roads, and destroyed forest, increase the agricultural irrigation, land usage for the agriculture is increased. All they put a pressure on the water demand. So that affects the quality of water, quantity of water, and the healthcare system. So this increase the prevalence of water-related disease. So this is other change. I mean, some of the challenges. Then I will concentrate on one particular issue specifically. So water is a vehicle for the transmission of most of the pathogens, and it is a key or important vehicle for the transmission of viruses, which are responsible for the diseases of gastrointestinal tract. Gastroenteritis due to virus is a severe problem in most of the country, affecting young children below 5 years. They are affected. So these viruses, especially the polio virus and other enteroviruses, they are released in the patient's stools for many days at high concentration. So once a person is infected with any of these viruses, he used to continuously release these viruses in large quantities for many days. So this have a chance to get mixed up with the water bodies, and that destroys the uh, waters available for drinking and consumption purpose, and even the fruits and vegetables may get contaminated with water. When a water which is not pure, a contaminated water used for the washing of fruits and vegetables, that may lead to spread of this virus. So that is a problem in this case. So waterborne diseases, especially viral diseases, are serious problem causing epidemics throughout the world, and this is a problem. And mostly the viruses of this particular group, they are resistant to the environment in the intestine. When these viruses enter into the intestine, they are able to survive there. Intestine is a harsh environment for most of the microorganisms, but still some of the viruses, especially this group of viruses which cause intestinal disease, are resistant. So that is a problem, and they are have a strong capsid molecule free of envelope. Non-enveloped viruses still pose a problem because they have a strong protein coat which gives protection for their viral genome, and most of the methods by which you either inactivate or kill viruses by destroying their genome. And if they have a strong protein coat, there is less possibility of attacking the virus. And vaccination is the only possibility in controlling the spread of waterborne pathogens and thereby controlling the waterborne diseases. But vaccination is not going to be that much easy. Of course, microbiologists are studying about a lot of pathogens, studying them in detail, but still vaccines are not available for many of the diseases. So vaccination is hampered by some factors. One is there is no effective treatment for most of the viral diseases. So what you have to do is you have to depend on the vaccination. Second, the viruses have a large epidemic potential compared to other organisms. Of course, we are seeing in the case of coronavirus, no bacteria except that what you call the flu virus and that plague, except the plague. This is a serious viral disease which is being a pandemic. So they are causing pandemics with a lot of epidemic potential. So that is a major problem. You have to develop vaccines. And what is hampering the development of vaccine is the genetic variability of the viruses. Everybody knows keep on changing its genome. So evolving into new serotypes. So vaccine development is not possible. That is why most of the death in under 5 children are due to enteric viruses. Then globalization is also having a negative impact because people used to move. Of course, that is the case in the case of coronavirus. Started in one country, spreading to all other countries because people are moving from country to country, carrying their luggages, animals are brought, goods are brought. Everything is a carrier of virus or carrier of pathogen. So globalization is also having some negative impact, increasing the emergence of new pathogens in new areas. So that is how these waterborne pathogens are posing challenges in controlling them. So I want to close this talk by asking you a question. Can you tell which of these water glasses is life-threatening? So think before you drink. Thank you. Thank you.
Yes, sir. Uh,
Yeah.
can you hear me?
Yes, ma'am.
Yes, we have few questions that can be answered. There are so many questions in the chat box. Maybe answer
Okay.
for a few. And I've also sent them an email ID so that they can post their queries, if any, in the mail so that we can answer
Okay.
them.
Definitely, ma'am.
So,
Yes, ma'am.
yes, I'm going. I'm going. Yeah. There are so many good morning, good evening, thank you messages. So it's, yeah.
All of them a very
What
nice
are,
good morning. It
uh,
is good afternoon now.
yeah. So it is a question from Tani Daspal. What are some of the more concerning emerging pathogens of concern which we have limited knowledge of? I repeat the question. What are some of the more concerning emerging pathogens of concern which we have limited knowledge of?
Okay. If the question is related to waterborne pathogens, we know what are the frequent waterborne pathogens which are causing problem, but still, especially as I told in the last few slides, viruses are the real challenges because we know the characteristic of the virus, but once it is keeping on changing its genome or its characteristic, it is very difficult. One. Second, there are a lot of new microorganisms being found as pathogens linked to water. Okay, I have not mentioned it here. There is a disease by name tularemia, which is now an emerging disease, especially through water, spreading through water, irrigation water. That is an area where these waterborne pathogens are becoming a serious issue. That is, irrigation water has been linked to waterborne diseases. Mostly, we think in terms of water used for human consumption, drinking water. So, a lot of organisms are there, and even the organisms known already. They have been reclassified, and when you are going for new techniques like PCR, you have to update the techniques, and accordingly to the available modern techniques, you have to go in depth about the organism. And parasites are becoming a major issue. I have not quoted here. A study between 2011 and 2016 says most of the waterborne diseases in a few countries are because of parasites, not because of virus, not because of bacteria.
Okay, so shall we go for the next question?
Please, ma'am.
Is there any relation with COVID-19 and waterborne pathogens? A question raised by Dr. Isabella Rosalind. Is there any relation with COVID-19 and waterborne pathogens?
Waterborne pathogens, as of now, it is not linked to corona virus or COVID-19. But when you talk about the comorbidity, of course, there are so many diseases which are very serious, like blood pressure, high blood pressure, heart problem, and diabetes, which are actually considered as comorbidity. But it is not like you take the example of AIDS, which is actually having a play with the immune system. So there it becomes severe. But as of this, yet we are not able to directly link COVID-19 and waterborne diseases.
Yeah, thank you, sir. There is one more question by Tuhin Jana. Which method, you have explained so many methods of detecting waterborne pathogen, which method of detection of pathogen is best?
If it is affordable, PCR, because it will be very up to the point. You just look at the gene and identify it. So that will be the method. But still, and here the problem is, as I explained in the talk, we have to find out whether there is a possibility of presence of waterborne pathogens. I think you can understand it clearly. Whether there is a possibility of presence of waterborne pathogens. That is a main aim, because you have to purify the water, or you have to make the water free from contaminants. The first method is looking for an indicator organism. So if you are looking for the indicator organism, that is enough. Then if you want to diagnose it clinically, then you will be looking for the organism. So PCR is the best method, and now there are sophisticated methods, because as I told you, the difficulty with all the methods, membrane filtration, MPN, they are good in detecting the possible contamination, but it is a laborious process as well as time-consuming process. PCR and other novel methods. It depends. What you need is you need instruments, sophisticated instruments, and you should be affordable to you. Affordable for the reagents. Kits are available. You can do.
Thank you, sir. One more question by Snehal D. Sir, excess chlorination of water alters taste of water and have marked for reporting symptoms like bitter taste of water or sticky film formation under the vessel. Can you suggest something on that? I'll
Okay.
repeat.
Yeah. I'm. This is one area I have not mentioned in the talk, because everybody says that the simple method, even for a public health authority, is to provide safe water is simply putting the bleaching powder or chlorination of water. But this is actually becoming a serious issue nowadays. There are two reasons for this. One, of course, the person has quoted the taste. Okay, it is nothing, it is more serious when you compare with other possibilities, because taste is a minor issue. But the problem is chlorine resistance among the microorganism is one problem which is being studied nowadays. Second, chlorine byproducts. Chlorine byproducts are more dangerous in some cases than the pathogen itself. Chlorine byproducts, when you apply large amount of chlorine, because of course I'm not finding fault with any system, but the technician or the healthcare worker who are putting the chlorine or chlorinating the water body, they are not keeping the proper concentration. Just like that, chlorine is used whenever there is a possibility of outbreak. Excess of chlorine is added. That creates a lot of other health problem. So chlorine taste, you have to bear it. Instead of diarrhea, you can drink a tumbler of water which is little bit bitter in taste.
Okay, sir. I think we can go with one more question and close the session. A question
Yeah,
by
please.
Shruta Srini Hazarika. It is a question by Shruta Srini Hazarika. Sir, when we boil the drinking water provided by the water vans, it is likely to form precipitates in the bottom. What could be the reason for this?
Okay, it is a practical question, because now, as I said, when we are not able to get a good water provided by authorities, we have to depend on private agencies. Many of the private agencies are not properly treating the water. Simply, they collect any surface water which is looking pure and adding lot of chemicals. Okay, I'm not very sure of it, but I came to know that there are chemicals which can make the ordinary water looks like a mineral water. The taste will be changed if you put a few drops of some chemical. So if they are putting a lot of chemical to change the taste and make it safe, that chemicals will be in excess in the water, and when you boil that water, definitely whatever is there will get precipitated. So that chemical, too much use of chemicals, some of them are not safe in nature and not safe in quantity. So that chemicals add to the precipitation problem when you boil it.
Okay. Uh, Thank you, sir. We shall end the session. The participants who have any more queries, I have typed the email ID to which you have to send the queries. You can just send it so that I can forward to Dr. Danny sir so that he can give you an answer for the questions that you are going to raise. We will close the session with a vote of thanks. Shall I proceed, sir?
Please, ma'am, any queries I am ready to answer if it is from students. I'm a teacher. I'm happy to answer the questions from the students. Of course, uh, if it is a faculty, I will share my knowledge. Thank you, ma'am.
Thank you. So we will go to an simple vote of thanks. Gratitude is a power catalyst for happiness. It is the spark that lights a fire of joy in everyone's soul. On behalf of the Department of Microbiology, Auxilium College, I deem it a pleasure in proposing vote of thanks at the end of the session. First and foremost, I sincerely thank the Almighty for making today's webinar a resounding success. I extend my gratitude to our invited speaker of the day, Dr. Joseph C. Daniel, Associate Professor and Head, PG and Research Department of Microbiology, St. Joseph's College of Arts and Science, Cuddalore, who spared his valuable time from his busiest schedule to share with us his expertise. Today, we had an opportunity to listen to you, sir, and thank you so much for the very informative and interesting presentation. I'm immensely grateful to Reverend Sister Alice Katie, the Secretary, Dr. Sister Jayashanti, the Principal, Sister Jacinta and Dr. Sister Amla Valarmati, the Vice Principals of Auxilium College, who always motivate and bring best out of us by all means. I thank all the staff and students of the Department of Microbiology for supporting me during this crucial period in imparting and exchanging knowledge. A special round of thank you to you, dear Ms. Deepa, Assistant Professor of the Department of Microbiology, for her technical support in bringing up this event into reality. This day would have been meaningless without the presence of our successful learners. Yes, my dear participants, we were amazed to see you register in such great numbers from various places in and around the globe. Even though it is a national webinar, you made it solid international. Thank you for your overwhelming response. We have connected today with participants from Pakistan, London, USA, Switzerland, New Zealand, Zambia, and South Africa. And of course, my own beloved Indians. Once again, I thank all of you for your cordial cooperation and expecting the same in the future endeavors as well. Thank you and have a great day.